Hello everyone. So welcome to another session in biochemistry. Here we are going to look into in detail the conformations of DNA duplex. Okay. In the previous presentations, we looked into the structure of DNA, how nucleotides were formed from the phosphate, the pentose sugar, ribose or deoxyribose, and the purines or pyrimidids. Right. We also saw how the nucleotides are bound together by phosphodiester bonds, and how a polynucleotide chain was formed. In one of the previous presentations, we looked in detail the structure of the DNA duplex, the double helical structure of DNA, as explained by Watson and Crick in their model for DNA structure. And the Watson and Crick model, it was based upon the DNA diffraction studies conducted by Rosalind Franklin and Maurice Wilkins, and the observations made by Edwin Chargaff, as stated as Chargaff's rule. So here we are going to learn about the DNA conformations. The different conformations of DNA, it is actually associated with the different conformations of the nucleotides of DNA. Okay, so they are associated. The difference in conformations, obviously, it is going to affect the DNA conformations. Conformations of nucleotides, as they change, the conformations of DNA is also getting affected. Apart from that, the physiological conditions can also influence the conformations. Now, what are the major DNA forms that exist? The DNA duplex can exist in at least 6 different forms, that is A, B, C, D, E, and the Z DNA form. And here you can see the A, B, C, D, and Z DNA. Here you can see the A, B, C, and D, they are right-handed structures. That is, they are right-handed helical, have a right-handed helical structure, as against the Z DNA, which is left-handed one. Okay, so the Z DNA, it is unique in having a left-handed helix pattern. Okay, now we will see what are the other important features of the various DNA forms. This particular table, it gives the characters of the A, B, and Z. It is comparing these three different conformational forms of DNA duplex. The A DNA, it is the broadest one, in the sense among these three, you can see here the helix diameter is 26 angstrom. Specifically, it is 25.5 angstrom. So what happens is, as against B form and Z form, the A form is the most broadest and the widest. Okay, so it is shorter but wider as compared with the B and Z. Okay, and you can see here it is having 11 base pairs per helical turn, and among the 11 in a single helix, you can see it is 2.3 angstrom. The adjacent base pairs are placed 2.3 angstrom apart. Okay, so 11 base pairs per helical turn, and the adjacent base pairs being placed 2.3 angstrom apart. Then, as already mentioned, it is 26 angstrom the helical, the width, or we call it as a diameter. Okay. Apart from these parameters, you can see that the major groove of ADNA, you can see here, this is the ADNA. Okay, the major groove of DNA is deep, but it is narrow. So what happens is, the base sequences or the information content of the major groove remains inaccessible for proteins. So the proteins cannot access the information content present in the major groove because it is very narrow. On the other hand, the minor groove is wide. It is shallow, but it is wide, and hence it gets accessible to the proteins. The information content present in the minor groove, it is accessible to the proteins. But the information content in the minor group, it is far lower than what is present in the major group. Okay. Now, when can we find the A DNA forms? Even though it is not so common, the A DNA form, it is usually favored at lower water concentration. When the system has a lower water concentration, the A DNA form is actually favored. Okay. Now, we can see the B DNA, how it does. B DNA is the most common form of DNA. And actually, Watson and Crick, when they explain the structure of DNA double helical structure, Actually, it was of B DNA. Okay, so all the parameters which were mentioned during the explanation for the double helical structure of DNA, it was of the B DNA. Here you can see it is 20 angstrom. It is having a helical diameter of 20 angstrom. There's a helix width. We are speaking about this particular distance. Okay, this length. It is actually 20 angstrom, and there could be 10 plus base pairs per helical turn, and the adjacent base pairs being placed 3.4 angstrom apart. Okay, the pitch per turn of helix is 35 angstrom. That is a single helical turn. If you take this length, it is 35.4. Okay, as against 25.3. So as compared to this one, the A DNA, it is more elongated. Okay, it is more elongated, but it is narrower as compared with A DNA. Okay, it is more narrower and it is elongated. Now you can see we have already seen all these parameters when we studied the Watson and Crick model for DNA helical structure. Here you can see it is 35 angstrom, the pitch per turn of helix, and 10 base pairs per helical turn, and the adjacent base pairs being placed 3.4 angstrom apart, with the helical diameter 20 angstrom. Okay, so what are the other parameters? You can see the major groove. Okay, major groove is wider and is more accessible to proteins, while minor groove is narrow. And usually the B form of DNA is favored at higher water concentrations. Okay, it is the most common form, but it is favored at higher water concentration. Now regarding the Z DNA, as against A and B, the Z DNA, it is left-handed helical pattern. Okay, the Z DNA, it was discovered by Alexander Rich in 1984 when Rich was solving the structure of CG repetitive sequence. Okay, so a stretch with repetitive CG sequence, how the structure could be. When Rich was studying the structure, actually he discovered the Z DNA form. Okay, so what does it indicate? That is, Z DNA is the form, it is favored by high salt concentration. So system with a high salt concentration, usually the DNA will try to be in a Z DNA conformation. And the Z DNA conformation, for that to happen, it requires alternating purine pyrimidine sequences. So that is a must. That is a stretch, a base sequence with purine pyrimidine alternating sequences can very easily exist in Z conformation, Z DNA conformation. Okay. Now, what are the parameters? As you can see, it is just helix diameter just 18 angstrom. Helix diameter means this is this length. Okay. So this particular length, it is just 18 angstrom, as against 10. You can see here the 20 and 26 in the B and A respectively. So it is the narrowest one. Okay. And what about the pitch per turn? So what does pitch? Pitch means the length of a single helical. This one. Okay. Starting from over here till here, this is the pitch. Okay. So what is the pitch? You can see pitch is 45.6 angstrom, as against 35.4 and 25.3. So it is most elongated one. Okay. So when we see this structure of Z DNA, it is a most narrower and elongated than the A DNA and the B DNA. Okay. Now there could be 12 base pairs per helical turn in the case of Z DNA, and adjacent base pairs are placed 3.8 angstrom apart. Okay. Now, what are the other features? As already mentioned, Z DNA conformation is usually favored at a higher salt concentration, and when there is alternating purine pyrimidine sequence in the particular stretch. Okay. Now, the major groove here, it is not prominent, and minor groove, it is narrow. So Z DNA is actually very narrow and very closed conformation. Okay, so this is about the A, B, and Z DNA. Right. Now we'll see the next. Okay, the next concept, it is denaturation of DNA. So what is denaturation? As we have already seen, the two polynucleotide chains, they are bound by hydrogen bonds between the complementary bases present on the two separate polynucleotide chains. Okay, we can see that this double helical structure, it is usually maintained, or it is usually stabilized at a normal temperature. That is maybe around like 55 or 60 degrees centigrade. Usually this double helical structure is stabilized. But when the temperature of the system is increased to about like more than 75 or almost like 90, what happens is the hydrogen bonds between the polynucleotide chains tend to break. Okay, it gets disrupted. Okay, it gets broken, and this will lead to the separation of two polynucleotide chains like this. Okay, and this particular process is known as denaturation or annealing. It is known as the annealing. Okay, so the denaturation is simply the loss of the double helical structure because of the disruption of hydrogen bonds, or separation of the polynucleotide strands due to disruption of hydrogen bonds. And this can happen when there is an increase in temperature, or when there is a change in the pH present where the DNA is found. Okay, but you can see over here, when the temperature is brought back to the normal condition, these complementary polynucleotide chains can come together and actually can form the DNA double helical form again. Okay, it can provide the DNA duplex condition. Okay, so this process is known as renaturation. So renaturation, it is actually the process in which the separated complementary polynucleotide strands, it can come back and form the DNA duplex. Okay, so this is renaturation, or it is otherwise known as reannealing. Okay, reannealing. Next concept we have is the melting temperature. Melting temperature, or TM, is defined as the temperature at which half the helical structure, half the DNA helices is lost. In the sense, if there are, just imagine if a system is having like 10 DNA duplex, okay, 10 this is what is known as a DNA duplex. Okay, I hope it is clear to you. So if a system is having 10 DNA duplex, and if at a particular temperature, when the temperature is increased, just imagine the system, the temperature of the system is getting increased. Okay, we are increasing the temperature of the particular system. At a particular temperature, we find that 5 DNA helix has lost its double helical pattern. Okay, and it has got separated as two polynucleotide strands. Okay, so out of 10, 5 has lost the DNA double helical structure, and that particular temperature is known as the melting temperature. Okay, it needn't be always like 5 out of 10. It could be even this part of the DNA getting separated, while the other part is still remaining in the double helical pattern. Okay, so it could be in that way also. So it is a temperature at which the half of the helical structure of DNA is lost. Okay, and this depends upon the nucleotide sequences present in the particular stretch. When there is a high amount of GC content in the DNA, the melting temperature increases. Why it is so? Because there could be only two combinations, isn't it? One is GC or AT. Between the G and C, we can find there are 3 base pairs, 3 hydrogen bonds. While between AT base pairs, they are bounded by only 2 hydrogen bonds. And hence, what happens is GC base pairs are more stable. They are more, strongly bonded. And to break that bond, the 3 hydrogen bonds, the temperature required is more. Okay, so in those cases, that the melting temperature, it is greater for DNAs with higher GC content.